Good afternoon, everybody. Thank you for joining us for this live event for the Southampton Science and Engineering Festival. So my name's Robert and I am a postdoctoral researcher in the Centre for Cancer Immunology.
And I am Michaela Christodoulaki, and I'm also a PhD student in the Centre for Cancer Immunology and doing my PhD in cancer sciences.
So as I said, this event is part of the Southampton Science and Engineering Festival. So this has been running all week and continues through until Sunday. So there's a load more events going on, including another couple of events after we finished today. So please check out the website to find out more about the other events that are carrying on throughout the week. So this event has been organised by the Wessex Immunology Group, who are a regional group of the British Society for Immunology. And this event is all about COVID and the role that it plays in the immune system. As well as the live event today, we also have a Padlet site. So this is a website that contains loads of information about COVID. So it's got scientist profiles on there, so you can find out about the research going on in Southampton. There's also some videos on COVID and the immune system. And for those of you who've attended the Science and Engineering Festival in person, you'll know that the WIG group tend to do making your own snots as part of that. And you'll be pleased to know that the instructions on how to make your own snot are also on that Padlet site. So hopefully the link to that will be appearing in the chat, so you can check it out throughout the event or afterwards. So for tonight's event, we had a brief video to start off with on the immune system, and then we're going to hear out from 3 experts about COVID vaccines and treatments. So hopefully these are the ways that will mean that this time next year, we'll be having the Science and Engineering Festival in person rather than online. But nonetheless, this proves to be, hopefully will be an interesting event. What we'd really like to encourage you to do is to ask your questions. So after each speaker has presented, there'll be loads of time for your questions to be put to them. So if you type your comments throughout the evening in the chat, either on YouTube or Facebook, and we will put as many of these as possible to the experts. So there's no such thing as a stupid question. Anything goes, and we will ask as many of them as we can.
Yep.
So first up is a video that's been created by 2 undergraduate students at the university, so Antonio and Matthew. And this video gives an overview of how the immune system is affected by COVID.
Coronavirus is a respiratory virus that is carried in droplets. When an infected individual coughs or sneezes, these droplets will be expelled, travel in the air, and be inhaled by a member of the community. When in the trachea, these droplets will break down, and the viral particles will travel from the trachea all the way down into the alveoli. The viral particles will enter the alveoli and infect lung cells. We're going to just quickly have a look at how coronavirus actually enters your cells. On the surface of the virus is a protein called a spike protein, which is sort of like a key. And on the surface of the lung cell is a receptor called ACE2, which can sort of be seen as a lock. It has to be specific in shape and match the shape of the spike protein. The spike protein binds to the receptor, and upon binding, the lung cell will take up the viral particle. When a normal virus infects your lungs, they bind to specialized sensors on immune cells. These immune cells are called macrophages and dendritic cells. Upon binding, these cells become activated and they raise the alarms in the lung cells and release specialized protein molecules called cytokines and interferons. The interferons communicate with surrounding cells and say, hey, look, there's an infection. You might want to put up some barriers and guards. Any subsequent virus that enters the lungs can't infect these cells. The cytokines serve the function of recruiting specialized troops into the lungs to help fight the infection. These cells move from the blood into the lungs and require activation from interferons. The immune cells will travel to the site of infection where they will destroy the virus. When a cell is infected with a virus, pieces of the virus end up on the surface of the infected cell. This immune cell, called a T cell, will bind to this fragment of the virus and kill the infected cell. This immune cell, called a B cell, will release antibodies against the virus, which prevents the virus from entering the cells and allows destruction of the virus. When the infection is cleared, B cells will divide to produce memory cells. These cells remember what the virus looks like, so when you're reinfected with the virus, it has no opportunity to infect any cells. So that begs the question, what makes coronavirus different to normal viruses? Well, only cytokines are released by these immune cells. No interferon is released. This means nearby cells can't put up any barriers to infection, meaning coronavirus can freely enter these lung cells. The cytokines released will still attract immune cells from the blood, but there is no interferon to activate these immune cells. They'll travel to the site of infection, but their ability to destroy the virus is severely compromised. The spike protein will end up on the surface of the infected cell, but the T cell's capability of killing the cell is severely reduced. The B cell is unable to release any antibodies, and the virus enters cells unchallenged by the immune system. The virus replicates unchallenged by the immune system. The replicated viral particles spread throughout the lung tissue, swarming the immune cells. This leads to overactivation of the immune cells, the dendritic cells and the macrophages, which leads to overproduction of cytokines. The cytokines attract more immune cells into the lungs, driving inflammation. The inflammation, coupled with the immune cells, causes damage to lung cells. They also cause blood vessels to become leaky, which causes fluid to enter the lungs. There are two types of vaccines for coronavirus. One is an adenoviral vaccine, which is the AstraZeneca Oxford vaccine, and the other is an mRNA vaccine, which is the Pfizer Moderna vaccine. Let's have a look at how these work. The adenoviral vaccine uses a dead and inactivated adenovirus, which carries the instructions for expressing the coronavirus spike protein on its surface. The spike protein is expressed on the viral surface, which is then targeted by cells of the immune system, which then become educated and trained to target that protein. With the mRNA vaccine, it's the mRNA itself that contains the blueprints and instructions for our own cells to make the spike protein. It enters our cells but cannot enter the nucleus where the DNA is. Our cells produce many copies of the viral spike protein, and our immune cells become trained and educated in targeting this spike protein and will know how to attack it when it encounters it again. Antibodies are produced and the viral spike protein is cleared. The mRNA gets degraded and doesn't remain inside our cells. Because the spike protein isn't coronavirus, only a part of it, you don't get the severe symptoms associated with the virus. When infected with coronavirus, you'll already have trained and educated cells to target the virus, memory B cells releasing antibodies. And hey, if one gets lucky, memory T cells will target that cell and destroy it.
Great, that was a very good video. Thank you to our students that created this. I would like to introduce Professor Saul Faust, who is a professor of Paediatric Immunology and Infectious Diseases within medicine at the University of Southampton and director of the Southampton NIHR Wellcome Trust Clinical Research Facility. He's currently focusing on delivering COVID-19 vaccine trials, stressing the importance of vaccines to beat the virus. So thank you for being here with us today. We are very happy to have you, and please show your screen.
Can you hear me and see me?
Yes, we can.
Great. Okay, well, thank you for inviting me. So I'm going to tell you a bit more about how the current state of where we are with COVID-19 vaccines. We've just had a really nice introduction from the video, and that makes my life a bit easier if I can get the slides to move on. I don't need to tell you this because you already know it. You've seen on TV and on the video you've just seen the coronavirus with the spike and how that gets into the body through the ACE receptors, and then the body's immune system has to respond by making antibody and creating T and B cell immunity. So the B cells remember how to make antibody, and the T cells actually fight the infected cells themselves. But the antibody provides that immediate protection for individuals by being able to stick to the spike protein. Now, when we invent a vaccine or when we develop a vaccine, we need to know lots of different things before we can start using them in the population. We need to know that they're safe. We need to know about the immune response the vaccine generates, whether that in itself protects against infection, how long the protection lasts, whether there's a risk of enhanced disease with the vaccines. This is important because some previous SARS vaccines actually caused a problem, but there's no sign of enhanced disease with the current COVID-19 vaccines. And we also need to know whether we're going to need boosters, how long the immunity is going to last, or whether we're going to need to combine the technologies in sequence, what we call heterologous prime boost. That just means using one vaccine and then a different one instead of using 2 doses of the same vaccine. So in fact, there's lots of different types of vaccine being developed, and you've heard a little bit about 2 of them, but the UK has bought vaccines in 4 buckets, as the Vaccine Task Force have described them. We've got the replication incompetent vector vaccines. This is the Oxford vaccine and Janssen. So although these are not actually dead, they're modified, so they can't replicate in the cells of people, but they can express the spike protein. You've just heard about how that then generates the immune response. So these are common cold viruses. The Oxford one is a chimpanzee cold virus, and the Janssen one is a human cold virus, not actual coronaviruses themselves, that have been modified to express or show the body that spike protein. The RNA vaccines are pieces of RNA. This is like a Snapchat message for the body's cells, a piece of nucleic acid that is just makes its protein and then disappears. And these are the Pfizer and Moderna vaccines, and they're delivered to the body in a package of fatty material called lipid particle that then can also help stimulate the immune system. More traditional technologies are Novavax and Medicago. The Novavax study is just waiting with the MHRA for formal approval. We think it's going to be approved soon because you might have seen that data in the public domain. Another very good vaccine in the clinical trials. And the Medicago clinical trials will start later on in March or April. Valneva, we've done the phase 1 trial in the UK. This will also have another phase 3 trial that's comparing Valneva to the Oxford vaccine starting in April. And this is an inactivated whole coronavirus, but it's been killed. This is the killed vaccine. Very traditional vaccine technology. We know a lot about how these vaccines work. And the reason why this vaccine might be good is because a whole vaccine doesn't just rely on the spike protein. It's got the whole coronavirus. So it might actually be that we can protect against mutated vaccine virus using the inactivated whole viral vaccines. So this is the pipeline, as I've just been describing. There's another RNA one that might come to the UK called CureVac as well. And you'll see some of the vaccines we've trialed have fallen by the wayside at their early phases. They're not going to come through in the near future, though they might come back in future when they've been modified. So this is we got involved in these vaccine trials right at the beginning in April. We took part in the phase 1 Oxford trial that started recruiting in April and May 2020. We had to do this in the sports hall at the University of Southampton. You can see, to manage the social distancing, you can see people going around the room, watching a video, meeting the doctors and nurses, having their blood tests and vaccines. People really taking part was fantastic to do that on behalf, taking a responsibility and a risk at that point in time to put themselves forward as volunteers for us to be able to develop these vaccines. And then the whole vaccine program expanded, and we did this via a national network run by the National Institute of Health Research, where every region put all its infrastructure together, and we collaborated between the hospitals, and the different bits of research money were given to run trials in the hospitals and the NHS and universities. And we managed this in Wessex by joining up Southampton, Bournemouth and Portsmouth. And we had 3 locations, we've got 3 locations now. We've been managing with Southampton and Bournemouth, and a new one in Portsmouth soon. And we've together done the coordination and governance and staff training, and managed the physical location logistics, and having 24-hour on call for participants who become unwell. And we have to monitor subjects for infection, because one of the things the Oxford trial did that the other trials haven't done is actually do a test every week to see whether the vaccines can reduce transmission and carriage, as well as being good at protecting against coronavirus disease. So we did this across the whole system with help from the NHS and the universities for this activity. So what do we know now? Well, we know that all the vaccines prevent hospitalization and death. The Oxford one so far is the only one to demonstrate real effect on asymptomatic carriage, i.e. transmission. That's the bugs we carry in the back of our throats. And you can't compare the trial data directly. All the trials have been done slightly differently with slightly different definitions of COVID. And although the Oxford and Janssen trials show lower headline immunity, actually cases were included with mild symptoms, whereas the Pfizer and Moderna studies were much stricter. And we also know that the adenoviral vectors vaccines are known to build immunity with longer interval between doses. This led, with the initial data that was available from both the Pfizer and the AstraZeneca Oxford vaccines, to the JCVI, the Independent Scientific Committee in the UK, deploying the vaccine first to the priority groups in whom 99% of death was expected. And they also changed the dose interval to 12 weeks to give the first dose to provide protection for as many people as possible, to try to reduce the pressure on the NHS and the intensive care units. One of the studies that led to these decisions, we actually enrolled all 240 over 70-year-olds in the phase 2 Oxford trial in Southampton, as just before the main recruitment to the main phase 3 trial. So, of course, when we get to the December and the regulatory agencies, we're seeing this data in real time. Clinical trials are normally done much slower, but no corners were cut. Everything that is normally done, including tens of thousands more people in each trial than would normally be done for new vaccines, was done in parallel, in real time. And the MHRA in the UK, actually, one of the few things I agree with Matt Hancock on many occasions, is actually the MHRA are global leaders in medicines regulation, and they were working with the trial teams as an independent body overseeing the studies, and they had already initiated this rolling review of the data, so that when they were ready, when the initial results came in, they were ready to be able to approve or not the vaccines that were submitted. It's been a huge disappointment that politicians and scientists have made statements in Europe and in America without understanding the data or process, and that's actually been a grave mistake. We've now got huge vaccine hesitancy in Germany, France, and other places for the Oxford vaccine, which I'm going to show you in a minute, is effective in the real world, as effective in the real world as Pfizer. Actually, we know from other programs like the Children's Flu Program that as the NHS program proceeds, people are likely to accept the vaccines to protect themselves and others. Assuming that we carry on successfully, people will be confident and come forward to have the vaccine. The media can help us by not focusing on that hesitancy. The oxygen makes low-level anxiety for the average person worse. And there is, though, a high concern that people from the Black and Asian minority groups in the UK and US are less likely to come forward, driven by social media and misinformation. None of the rumours that have been put out there on social media are true. We need clear, transparent communication from the government, the JCVI, and so on. Ultimately, we probably do need a bit of control and regulation of social media misinformation. Other things are much more controversial. Vaccine passports have the potential to be very divisive. And it might be that we have to introduce them because the globe and other countries introduce them. But you can see at the moment the government are just trying to avoid doing that, so that everybody's been offered vaccine before we have before we start making rules about people who have and have not had vaccine and what they can do. So what's the real world data? You've seen in the newspapers data from Israel and Scotland and Public Health England, but it was a really interesting paper released as a preprint, so that's not yet been peer reviewed or looked at by other scientists and agreed for publication, but the data is in the public domain that was announced last week. And this is a really important study because it records the patients coming into Bristol, 2 large hospitals, and they've estimated the effectiveness of 1 dose of the vaccines in the real world by comparing the immunization rates in people with acute respiratory disease, in people who have got positive tests and those who have got negative coronavirus tests. And they've been able to structure the analysis to control for time, because the Pfizer vaccine started earlier than the AstraZeneca vaccine, and they also have controlled for factors such as social deprivation and care home residence and sex. And this is really interesting data because it shows that the Pfizer vaccine for 1 dose is 71% effective in people who have got a median age of 87 years, so the most vulnerable of our society. But the Oxford vaccine is even better, 80.4% with a median age of 88 years. In fact, when you take into account, when you reconcile the data to cover the same time period, they're both identical. 80% real world effectiveness in the most elderly population. So the Germans got it wrong, the French got it wrong, the Americans are going to get it wrong because they're not going to allow enough time between their doses. And it just shows in a pandemic, it's really important to have a detailed understanding of the data and also take into account that the trials have had to evolve and change their design during the pandemic based on data that was made available as we went along. So in the future, we need to optimize protection. We need to work out the vaccine combinations, and there's studies going on. We're looking at children and pregnant women, at the immunosuppressed. We've got to look at the viral mutations and whether we're going to need to change the vaccines. And those studies will happen. The third dose booster study and mutation vaccine studies will happen later in the summer, before we get to the decision about whether the UK are going to have a third dose booster in the autumn. We've got to protect the global manufacturing supply chains for all the different vaccines. And somehow we've got to avoid this nonsense of vaccine nationalism. We're going to need all the vaccines to be good, because the whole world has to be immunized and keep on being immunized if we're going to keep the pandemic at bay going forward. So which vaccine should you have? Any deployed vaccine in the UK have been and will be fully evaluated by the MHRA. We don't have uncertainties about safety once the MHRA have given that regulation 174 approval. We do have uncertainties that we're still investigating about how long the immunity will last, how often vaccines should be given, and how well they're going to work in different age groups. The current schedule is evidence-based and aimed at reducing deaths and admission to hospital. We're going to be sticking with that 3 months for now. Please take the first vaccine offered to you. And thanks very much for listening.
Thank you very much for this presentation. It was great. I would like to ask you a question. Are there any risks or side effects of the vaccines?
So there's no special side effects of these vaccines compared to any other vaccine. Some people get a sore arm, some people get a little bit of fever, some people get chills, but generally these are very short, mild and long-lived. And you can actually look at the MHRA website. They do a rolling weekly summary of all the different side effects that are being reported. And there's no concerns from either the Pfizer or Moderna vaccine, Pfizer or AstraZeneca vaccines at this moment in time. There's a bit of a news piece today, isn't there, about blood clotting in Denmark, but that hasn't been seen in the UK so far. And you can absolutely be assured, everybody can be reassured, that the MHRA are looking very closely in continuous time at whether those signals are seen in the UK as well.
So to follow up on that, is there anything known about what might make someone more likely to experience a stronger reaction? Not necessarily a major kind of adverse event, but perhaps where some people get just a sore arm and others perhaps might have flu-like symptoms for 24 hours. Is there anything known about what might affect?
No, not at all. And interestingly, just because you've had a reaction in the first dose doesn't mean to say you're going to get it from the second. In fact, I think it's almost the opposite for some of these vaccines. So it is just one of those things that can happen. And if you have a really sore arm on dose 1, don't worry, it probably won't be as bad for dose 2, but we can't promise you that.
That's reassuring for someone who had a sore arm from dose 1.
I can see actually there's a follow-up question to that in the chat that Jessica's just put on there, and that's about if a single dose of vaccine protects, why do we need that second dose at all? And that's a really good question as well. The answer is because we think the second dose is going to provide that longer lasting and memory immunity. Many vaccines need boosters. In fact, the Janssen vaccine has now got approval in the US and probably will be approved in the UK in the next few weeks for a single dose vaccine. But we're also doing a trial of 2 doses in the UK because we need to know whether 2 doses of Janssen is going to be better for long-term protection. So it does look as if in some populations, like cancer patients and others, that the second dose is definitely going to be needed. But, you know, while we've got lockdown and social distancing, 1 dose getting into everybody is the right thing to do, and then the second dose at 3 months. And that's how we're going to take the pressure off the health service. So
there's a comment related to that that's just come in, basically asking if the Johnson & Johnson vaccine is different in any way from the ones that we've already had approved, or I guess is it just a simple fact that they designed their trial as 1 dose to start off with?
Oh, Johnson & Johnson covered all their bases. So they've got their technology is similar to the Oxford AstraZeneca technology. Their Janssen is a human cold virus that's modified, and they've got a licensed vaccine for Ebola based on the same technology. The Oxford one is the first licensed or approved vaccine they've got for this technology, though they've been working on MERS in the past. That never actually progressed because there was no need for it at the time. So they're similar technologies, not the same. Johnson & Johnson design their trial so that they could cover the bases of needing 1 dose or 2 doses. So that's why they've got these 2 big 30,000 plus people trials. Some people getting 1 dose and some people getting 2, and they will be able to then decide how to advise the regulators and vaccine committees all over the world how to use their vaccine to deploy.
We have a question from Anne. What do you say to someone who is not taking up the vaccine because they're worried that the vaccine is made from a chimpanzee viral vector?
I'd say, look, people have got all sorts of worries, but it's not, as the video showed before, this can't give you a cold. It's been taken apart and modified, so all that the viral vector does is it delivers. You deliver it to a muscle cell and it produces spike protein, and then the body clears it up and mops it up. It can't do anything else, and it's eliminated from the body. I think, people are worried. People don't generally read the medicine packets. They don't generally look at the small print on medicines given to them by the doctors. And so I think one of the things that social media has done is it's caused this rumor, this swell of complete nonsense and myths, all of which we have to answer and say, well, look, that's not true because there's no pork in these vaccines. They're not made from fetal cells. They can't do anything to your body. But, people can be confident. We've now done the biggest clinical trials ever for vaccines, and we've deployed the vaccine to millions of people, and we're not seeing any major side effects. So people should be fully confident that they can go and get a vaccine when they're called to by the NHS.
So another question that's come through from Emma, is there any evidence that the vaccine being less effective if it's being given to you when you've had COVID at the time? I know this is something that gets asked in the screening before you get the vaccine.
Yeah, so all the trials have been done without stopping people who've had COVID taking part, the big phase 3 studies. So between 10 and 15% of everybody who has entered one of the phase 3 clinical trials has already had COVID. So we know it's a safe thing for people to get the vaccine, and it acts as an artificial booster, if you like. We're not doing, if you're symptomatic, then you're sent away from you shouldn't be going to a vaccine center if you're symptomatic. But if you're just carrying it, then actually having the vaccine booster will not do any harm and is the right thing to do. Likewise, we did the swabs on people coming to the vaccine hubs, but we didn't act on them.
Great. Another thing is how long does immunity last with the vaccine? How much is expected to last?
We don't know. We've got no idea. That's why the people in the trials are still being followed. They still have blood tests every 3 to 6 months. Some of the studies are going to go for 1 year. They might get extended. Some of the studies are set up as 2. But we'll also be doing the booster studies. We're actually leading those from Southampton in the summer, where we're going to be inviting people who've received vaccine in December and January if they would like to come forward for a 3rd dose booster. And we'll have that information for JCVI and for the MHRA by the autumn, hopefully, so they can then make decisions on whether and who will need a booster in the winter this year.
I see.
So the next question is it unsafe to have the vaccine when you have health conditions such as tachycardia or thrombosis? No,
not. Again, the balance between getting COVID and having a condition that makes you more susceptible to being ill and receiving a vaccine, the balance is hugely in favor of having the vaccine. But what you should do is explain whatever medical conditions you've got to the people, the doctors at the vaccine centre, who will be able to give you specific advice, or if it's your GP you're having a conversation with, they'll know. If there is something about you that means they advise one or the other, like you've got severe allergies, then the doctors, either your GP or the person in the vaccine centre, are the right person to have that conversation with on an individual basis.
Okay, I think we have time for one more question. The people are asking whether it's possible to is the vaccine for children, is it safe?
Yeah. So we're doing the studies. So we've just started the studies for adolescents, for Janssen and for Oxford. So what we've got to do is make sure the dose is right. The vaccines are going to be safe in children, but we've got to make sure that the dose we give them doesn't make the side effects too big. And so that's what the studies we've started. There's no rush to immunize children because children don't get severe COVID. Very tiny numbers of children get an inflammatory syndrome, but actually we see very similar inflammatory syndromes with many infections. So we're in a pandemic and we're going to see more. This multi-system inflammatory disorder is new, but is very, very rare. So if we immunize children, it'll be when we've done the studies, which will probably be at not till the end of this year or beginning of 2022. And it really will be to try to help damp down the transmission. If we show the vaccines stop transmission between people, then we might use the vaccines to give to teenagers first and then maybe younger children later in order to reduce the chance of mutations. Now, when you've damped down the virus a lot in the population, like we're going to do with the vaccine, and it goes away in the summer, actually there's very little chance of mutations happening in children. So we don't have to get on with this quickly. We can just get ready for this if we need to do it in the next winter period. The whole point is to not have to go to another lockdown and to be able to stop other mutations in the future. But, you know, the last thing to say probably is we're heading towards a situation in the autumn where everybody in the UK, all the adults that have been offered a vaccine, hopefully most people will have taken it. And COVID will probably become just like another respiratory virus, like flu, where we'll get a vaccine once every year or two to boost our immunity. But we'll live with it because the impact on our hospitals is just like flu every year. A few people will go to hospital, but most people won't. A very positive future.
Very positive.
It's a nice positive point to finish. So I think in the interests of time, we'll move on. So thank you everybody for asking your questions. If there's any that haven't been answered, if you take a look at our Padlet site, then we will try to answer some of them on there. And thank you, Saul, for a fantastic talk and for answering everybody's questions.
No problem.
That was really interesting.
Thank you very much, Saul.
So now we're going to move on to thinking more about treatments for COVID. So we have two speakers. So we have Dr. Jonathan Martin and Professor Gareth Griffiths. So Jonathan is a senior house officer working in oncology, but he splits his time between working in the clinic and also working in the Southampton Clinical Trials Unit. And there he works on the design and management of clinical trials. Generally, this is focused on oncology, but more recently has moved into COVID-19. And Professor Gareth Griffiths is a professor of clinical trials at the University of Southampton and is also the director of the Southampton Clinical Trials Unit. So this is a unit within the university and runs a large number of international trials. Again, these have traditionally focused on oncology, but more recently have moved towards COVID for obvious reasons recently. So I'll hand over to Gareth and Johnny, and I'll let you take control.
Great, thank you very much. And this is, as mentioned, a duet between myself and Jonathan. So can I have the first slide, please, Jonathan, if you've got it? Has the first slide come up? I can't see the first slide. Hang on a minute, I'll see if I can share. Right, I will start it. I know Johnny was going to go through it, but I'll start and pass it to Johnny. So thank you very much. So the Southampton Clinical Trials Unit is actually based within the Centre for Cancer Immunology. And as you can imagine, in spring last year, suddenly cancer research sort of ground to a halt. So unfortunately, although we were doing lots of trials such as RNA vaccines in cancer, as we had a little bit of lull in cancer, we thought we'd turn our hand to what we can do with respect to COVID-19. So we're sort of going to present on the Agile national trial and some of the new immunological treatments that we're going to incorporate. So what is the Agile trial all about? Well, as soon as we got hit by COVID, there were a number of national trial platforms that were set up to look for new treatments. The ones you would have heard of quite commonly are Recovery, which are in the hospital setting, and also in principle as well, which is in the GP primary care setting. And they actually started off investigating a lot of repurposed drugs. So even now, recovery is looking, say, for example, aspirin as one of their candidates. The problem was when that started, though we were doing all that fantastic work Saul was mentioning with vaccines, there wasn't really a pipeline of new drugs that were coming in to be tested in these late phase platforms. And so even though some groups were doing preclinical, such as Porton Down up the road and other university-based researchers finding new targets and new interventions and treatments for COVID, there wasn't a mechanism in working out the dose and early signals of activity. And that's why we created the Agile, which is effectively the first in human and phase 1 and very early phase 2 to look at promising new candidates, so that the platforms such as Recovery and Principle and other platforms such as Accord, which we're running out of Southampton, can pick up those promising candidates. And basically, we designed Agile to encompass a master protocol that we can drop in multiple new drugs as they came along. And we can almost think of this as each of the drugs coming in with their own sub-protocols within the Agile portfolio, comparing against the current standard of care. So it didn't matter when they came into the study, and it didn't matter what the standard of care is, because what we were looking at is new drugs that we can work out the dose and the activity. It included both first in human healthy volunteers, but also COVID patients that were both in the hospital and the community setting. And it's a big ambition, so that, as Saul said, that in the future, when this becomes seasonal, that we're actually trying to prevent patients going into hospital. That it'll be a bit like when you get a cold and you might reach for the Lemsip. We're looking for all sorts of antivirals that you can take off the shelves at Mr. Boots to prevent you from even getting to hospital. I'm just going to cover some of the design aspects before passing to Jonathan. If I can get the next slide to come up. So this is how it works. We get a COVID patient, comes into the study. We consent them, we randomize them. They then start going through the dose selection processes, various doses, and they move through until we hit the optimum dose. We essentially follow them up until about day 29, which is the standard amount of time which we follow them up, or to at least then know whether or not there's activity within 29 days. Once we've determined that optimum phase 1 dose, we undertake quite a small phase 2, randomized phase 2 between standard of care. And at that point, we're saying we should drop that candidate because there's no evidence of activity, or it should progress to some of these larger platforms. Not wanting to dwell too much on statistics, as I'm a statistician myself, but we use quite a novel approach because we appreciate we can't use conventional designs if we want to act quickly. And we use something called Bayesian, where we actually develop a prior distribution. That's our beliefs of what the data might look like. We collect the data within the study and then actually work out a posterior distribution. And essentially what we're doing is we're determining a dose is unsafe if there's more than a 25% chance that a treatment gives more than a 30% higher dose limit toxicity over the controls, which is whatever the standard of care is at that time, even if new treatments have become the new standard of care. And then we keep accelerating or going up the doses until we actually find one where we accept about a 20% increase in dose limiting toxicity to take that forward. It then goes into a phase 2 where we use an approach where we look at hazard ratios, again using a Bayesian approach, but at various times we determine whether there's futility, there's no point for this drug to be taken forward later on, or whether there's clear signal of efficacy, so there's a signal of activity to take forward. And Jonathan's now going to talk about one of these candidates, so I'll hand over to him.
Hi there. Apologies for the technical faults. I couldn't seem to load my bit of the presentation. Gareth, would you be able to go through the slides as I talk through them? So yeah, I've been involved. So my name is Jonathan. I'm one of the clinical research fellows at the Southampton Clinical Trials Unit, and so we've got this trial platform that Gareth was talking about. We've been very busy indeed on this trial. So we've currently got 3 studies currently open to recruitment, and we've got multiple studies currently in development. So I'd like to talk to you about a study that I've been involved in for the better part of 6 months, which is the VIR study. So it's essentially the VIR study. So VIR 7831. So I'm just going to say VIR1 for short. It's a new antiviral drug being developed by GSK and VIR, and it's an antibody specific to COVID-19. And as you saw in the talk in the animation earlier on, the antibody actually binds to the spike protein on the virus and neutralizes the virus and blocks COVID-19 from entering the body. So this VIR1 was tested last year in 20 patients with mild disease, and it was considered safe. And there is an ongoing phase 3 trial of the drug. And actually yesterday, the interim analysis, so the kind of midpoint analysis, found that it showed an 85% reduction in hospitalization or death in COVID patients. So this is really exciting. So it blocks, it neutralizes the antibody and blocks uninfected cells from becoming infected. Next slide, please. So, and then VIR 7832, so I'll call this VIR2 for short. Again, it's a modified version of VIR1, and they've just essentially changed the structure ever so slightly in the antibody. And again, it blocks COVID-19 from entering the body. But what this modification we've seen in sort of monkey and mouse models, it's actually increases the ability to stimulate the immune system. So it neutralizes the virus like VIR1, but what it also does is it attracts immune cells such as macrophages and natural killer cells to come and attack and destroy infected cells. So that's kind of enhancing the antibody's ability to both neutralize and prevent infection, but also destroy the infected cells as well. Next slide, please. And the other exciting bit about this new antibody is, as well as its ability to stimulate the immune system, it also produces COVID-19 specific T cells. So from the talk at the start, T cells are really important for memory. So making your body remember that have had COVID, and what this potentially can do as well as it provides a treatment, but it can also provide and help with vaccine effect. So it works well with the actual vaccine, and it works as a treatment, but also a potential vaccine. So that's why we're really excited about this new antibody. Next slide, please. So where we are now. So the VIR study has opened to recruitment this month, so last week actually, and we've got a site open in Liverpool, and then we've also got a site open in about to have a site open in Southampton. So we're looking at the population we're looking at is adult patients with mild to moderate disease, so temperature, cough, but they're not in hospital. And the intervention, as I discussed, was either VIR1 or VIR2, and the control, or the placebo, or otherwise known as sort of the dummy drug, is saline, which is essentially salt water. So we know that VIR1 is safe and tolerable in patients with mild to moderate disease from the previous trial. But we also want to see if VIR2 is safe and tolerable. So that's what phase 1 is about. And once we find a safe and tolerable dose for phase 1, we will then move into phase 2. And phase 2 is to see if VIR1 or VIR2 is effective at treating COVID. So what we look at, what we will look at is symptoms, severity of disease, and time of symptoms. But we'll also look at the more sort of immunological things like a T cell response or other sort of immune related things to see if that modification has actually worked. And although there are treatments for COVID, there are treatments for COVID at the moment for severe disease when people are sort of hospitalized, but there's a distinct lack of treatment for early disease. So people that have mild to moderate symptoms. So we're hoping to prevent them from developing severe disease and ending up in hospital or on oxygen or on a ventilator. So VIR is just one of the 3 trials that are up and running, and we've got a lot in the pipeline, and it's just been a really exciting process, and it's a really good emphasis of how Southampton is leading the way, as you saw with Professor Faust for vaccines, but also for treatment. Yeah, so if there's any questions or comments, we'd be happy to take them.
So thank you very much, both of you, for that fantastic introduction to your trials. So if anyone has any questions, please feel free to write them in the comments, and we will read them out to Gareth and Jonathan. So to start with, you've spoken very nicely about the trials that are ongoing. So how do these compare to the current standard treatments that are being used on COVID patients in terms of what is the current standard of care?
So we know there's, I mean, I'm a statistician, but my understanding is, and I'm sure Jonathan will correct me, there's 3 effectively UK approved treatments, which is remdesivir, which is an antiviral, but that's used in the hospital setting. There's dexamethasone, which is an anti-inflammatory, as I understand it. Again, that's in a hospital setting, so quite poorly patients. And I believe there's 2, if I pronounce them right, salulimab and the toxelimab, which I think are monoclonal antibodies and immunosuppressive drug, which are done in quite critical sort of care. So these are drugs that, when you see the news and you see all those patients based in hospitals, they're using it that sense. There's no drug for when people like us in the community suddenly then get COVID, and within 5 days you start getting the symptoms and you go, oh my goodness, it's too late to get a vaccine. As Saul said, you're not allowed to get a vaccine at that point, and it's too late. What do I have? What can I head this off at the pass? And at the moment, there's no drug out there that's been approved, as my understanding, that you reach for, and that's what we're interested in. Although we're looking at the hospital setting, I think with the vaccination program going on, we should see, as we are at the moment, a lot less patients, and it's actually, I think where the treatments are going to work hand in hand with the vaccinations are those individuals that get the early community-based COVID, where they can take drugs so they never have to go to hospital.
Great. My question is, are these current treatments available? Are they available for both adults and children that we need to go to the hospital?
So these the ones that we're currently looking at are sort of first in humans. So they've some of these drugs have never been sort of tested before in humans, but we've got sort of lab data. So we need to pick our patient population really carefully. So for example, phase 1 is adults only between the ages of 18 and 55 with no medical problems, because we need to see the effect of the vaccine on a healthy individual. And if this is successful and goes through to the later trials, sort of phase 3 trials, where it shows you safety, tolerability, effectiveness, then you can start thinking about giving it to other populations. But the phase 2 will be sort of, the phase 1 I said was 18 to 55, but phase 2 will be adults 18 and over.
Okay, thank you.
So how many people are typically recruited to these trials at each phase?
They vary. It's quite a unique design, because we have this master protocol that specifies what the primary endpoint is, what the primary eligibility criteria are, and the statistical design. And what's happening is that we start talking to companies or scientists developing their candidate, and we have to sit down and basically go through the basics of PICO. What population do you want to recruit in? Because we're doing healthy volunteers, we're doing community-based patients, or we're doing hospitalized patients. You know, what is your intervention and how does it affect, and what sort of effect size do you think you do? And then we have to consider what the control arm is at that time, because if something's working really well, then you need sort of more numbers. And what the primary endpoint is it a safety, or do you look at virality? So it varies. We're finding in the phase 1, it could be anywhere between about 12 patients and about 20 patients to give us the optimum dose. What we're finding then in the phase 2, if it's in the community setting, it could run into about 150 patients, because actually we need larger numbers, because not all of those are going to go to the hospital setting. If they're hospital setting, then of course we can do it with less numbers in the phase 2, because you're looking at the time until they're discharged, you know, and there's you've got more statistical power. So per candidate, I'd say in the region of about 40 to about 150.
Thanks.
We have a question. Are there any side effects for our specific groups of people that might benefit from these antibodies? For example, people with lower immunity due to cancer or aging?
At the moment, we haven't included cancer patients. These drugs, many of these drugs at the moment are first in human, so we don't know. But what we do is we monitor them very, very closely. What we're working with is we've developed Agile with the UK Clinical Research Facility Network. Saul Faust is actually a director of our Southampton ones. And these are specialist units that do phase 1 studies. So the patient comes in, they're very, very monitored very, very, very closely. But we don't know a lot about these drugs. They may have just popped out preclinical labs, and it's the first time it's tested in patients, be them healthy or COVID. And we're collecting all that information. And we present that information about all those potential side effects relative to benefit to then make that informed decision about taking it into a much larger study. But we've seen this in the vaccine studies. You know, we know even when we're doing the vaccine studies, they would stop occasionally because they find some sort of side effect that's not expected. And within clinical trials, you look so closely about the safety and the populations, and you act on it immediately.
So one of the treatments that you mentioned that's been approved is dexamethasone, and obviously that was mentioned in the news quite widely. So how does that work to in COVID patients to reduce their symptoms and improve their outcomes?
Sure. So dexamethasone is again used in sort of severe settings when patients are either sort of intubated and ventilated. And what we see with COVID, what causes that sort of big respiratory problem is you get a massive immune response and cytokine release because of the virus. So dexamethasone is an immune suppressant. So giving dexamethasone at the right time in the right setting can dampen the immune system in the right way to prevent or make that effect less severe. It sounds quite contraindication, because it sounds funny because you're giving someone an immune suppressant to fight, a virus. But again, it has to be given at the right time and in the right setting to prevent that.
Great. Thank you very much, Jonathan and Gareth. Thank you all to thank you, Saul. Thank you to the students that made the video. It was so nice to have and answer the questions from the public too. And also thank you to the rest of the Wessex Immunology Group for putting this together. And thank you to the audience who came to this event. Please go to the Padlet and check out all the events that might be are left until the 14th for the Science and Engineering Festival. You can read more about how the immune system and COVID works, and also read more information about our hosts. Thank you very much for being here today. And please, check out the rest of the events until Sunday. Thank you very much. Please don't forget to tell us what you think of this event. If you liked it, here on the link on the page. Thank you all.
Thanks.